is from Sarath. So Sarath was trained like me in London, but he decided to move to Melbourne, and he's the director of pediatric respiratory and sleep medicine in Royal Children's Hospital in Melbourne. He's the head of the Department of Pediatric at Melbourne Medical School. He is a leading authority in infant lung function testing, and is regarded as a world expert in detecting early lung disease. So Sarath would be first talk is we'll be talking about early gut and lung microbiome. Does it matter? Thank you, Sarath.
Thank you very much, Atul, and thank you for those who are participating live and for those of you who are going to look at this video afterwards. It's also equally a shame that I couldn't come there and meet you, and perhaps even more disappointing for my elderly parents who are currently in lockdown in Lancashire over there in the UK. And I'm speaking from a very rainy autumn evening here in Melbourne. I'm not going to be talking about infant lung function, thankfully. I'm going to be talking today about the human microbiome and some aspects of that that might be of interest for respiratory pediatricians and perhaps also general pediatricians. The microbiome is obviously a hot topic. If you look at the number of COVID-related papers published this year, there's around 2,000, but 6,000 microbiome publications already in 2020, and I think about 15,000 last year. So it's an impossible topic to actually try and keep abreast of this, but I've done the best I can. Of course, the microbiome is that collection, the collective genome of bacteria, archaea, viruses and fungi that occupy our various niches within the human body. The estimated number that constitute the microbiome has varied over the years. We have an estimated 30 trillion human cells, that's 30 with 12 zeros, and about the same number of cells constitute the microbiome. There are somewhere up to 1,000 different species that occupy the human microbiome, but about 99% are taken up by around 30 or 40 of the most common species. So what I'm going to try and do in this talk is initially give a little bit of information about our understanding of the gut microbiome, because that's the easiest. The majority of those enormous numbers of publications have been directed, but also show some recent data in relation to cystic fibrosis and also atopic diseases, and concentrating, as the title suggests, on the earliest aspects of life and how the microbiome has significant associations in infancy. And we'll answer whether or not the microbiome actually has importance in terms of causality of human disease. So let's start with the gut microbiome.
Can I just step in? It's okay, don't worry, they are moving. Carry on.
So, we're going to answer this very quickly, and that is, does the early gut microbiome matter? Well, it does. It's been shown that the gut microbiome harvests energy from the food we eat. A fantastic paper by Trumpeter and Ben Marsland showed that it could actually help to entrain our immune system. We know that it breaks down foreign products and produces important metabolites. And there are also to show data that the human gut microbiome actually impacts the development of the enteric nervous system. So this is a fantastic slide from a very good paper by Kundu et al., which we've called the 7 Ages of Poo. When I was at school, I had the misfortune of having to study As You Like It, a pastoral play by Shakespeare. It has a very melancholic character in Jaques, who delivered a very famous soliloquy called the 7 Ages of Man. The first of those 7 ages was the infant mewling and puking in his nurse's arms. And so the 7 Ages of Poo starts with infancy, and that's the period where the gut microbiome is inoculated, mainly through the course of the delivery and the mode of delivery. So for example, for vaginal delivery, there's a certain inoculation of the gut microbiome with lactobacilli and Prevotella, whereas those infants who are delivered by a caesarean section have a gut microbiome that resembles more closely the skin microbiome with Staphylococci and Corynebacterium and Propionibacterium. The breast milk also forms an inoculant, and this is what feeds our gut microbiome with enormous quantities of lactobacilli and bifidobacterium. Over the subsequent 2 or 3 years, the early gut microbiome matures. There's a reduction in bifidobacterium and there's increase in richness. At puberty, the gut microbiome differentiates according to gender, which is certainly very interesting. And there's also an acquisition at this stage at the start of a resistome, a collection of genes that are associated with antibiotic resistance. In adulthood, that's the time when the gut microbiome is at its most rich and also its most complex. There are also changes that occur in the microbiome during pregnancy, particularly in relation to the placenta. Not represented on this slide is that there is a microbiome that's associated with senility. So there are certainly some gut organisms that seem to be associated with prolonged healthy life. So during the course of life, the microbial diversity and number increases within the gut, and the proportion represented by anaerobes also increases. So that's in health. And what about in disease? Well, in order to study this, most people have turned to the bacterial ribosome and the ribosomal RNA gene. So on the left here, you can see that the bacterial ribosome, this is the anvil on which messenger RNA is tied, and the tRNA binds to these, and you get these growing peptide chains. So it's really important in peptide synthesis. The bacterial ribosome has 2 subunits, a small 30S subunit and a larger 50S subunit. So when we look within the 30S subunit, there's a strand of RNA called 16S RNA, and then a lot of ribosomal proteins. When we say 30S, that refers to Svedberg units, which are actually measurements of sedimentation. This RNA, the 16S RNA, and also the 23S RNA that's in the larger subunit, are very conserved through all bacteria and also archaea. So the gene that codes for this has areas which are incredibly well conserved between all bacteria, but also some components which are variable. There are 9 of these. And traditionally, or most commonly, people, when they have done sequencing of this gene, have looked at the V4, the variable component 4. But there are others which actually confer an advantage when looking at certain microbiota. For example, the microbiota on the skin, when you're interested in the genera related to Staphylococci, it's probably best to use the V1 to V3 because staph have had some evolutionary changes in those parts of the gene. So what about disease? It's really impossible to look at all the studies that relate the gut microbiota to human diseases. But for those who are interested, I do recommend looking at this particular study by Duvalet et al. That was published a couple of years ago. In this study, Duvalet assessed 28 published case control gut microbiome studies that span 10 human diseases. And a remarkable exercise, they acquired all the raw data and the disease metadata from all these studies from all around the world. And they performed a cross-disease meta-analysis following a sort of systematic reprocessing and a complete reanalysis of all the data. So what they were aiming to do was to assess whether there were consistent and disease-associated specific microbiome patterns from across these multiple studies, and to see whether or not studies actually were replicated. What they actually showed was that many of the associations that are reported in relation to a particular disease are also shared in response to other diseases and also occasionally in health. This is sort of disappointing, but what it shows is when people report that there is a dysbiosis in the gut microbiome, this may not be specific to that disease, and therefore we have to be very cautious when we're looking to those patterns in order to develop interventions. Another useful thing that they did suggest in this study was a way of categorizing dysbioses that we identify in future studies. So for example, if the microbiota identifies bacteria or microbes that are associated with health, this may in future inform the composition of prebiotics, compounds given to promote the development of certain microbacteria, or probiotics. Another pattern which has been identified as a pattern of dysbiosis is where there is depletion of health-associated bacteria. And so this is obviously going to inform the interventions predicated around probiotics. Some dysbiota show dominance by pathogenic bacteria. Colorectal carcinoma is an example of this. And so in these conditions, there may be a causal relationship between the microbiome composition and disease. And so these might be better managed with approaches around antibiotics or even phage therapy. There are some patterns of broad restructuring of the microbial community. For example, certain diarrheal illnesses, and this is well known in relation to Clostridium difficile infection. So these are the conditions where microbial transfusions might be affected. So I mentioned we were going to talk about some of the recent data in relation to cystic fibrosis and other atopic diseases. So what's the rationale for studying the microbiome in CF? And we and others have studied, moving on from the gut, actually assess the lung microbiome in CF. Well, as many of you will be aware, lung function decline occurs despite our injudicious and on occasions use of antibiotics in CF. We know that even with the best treatment of exacerbations, about 25% have reduced lung function and isn't restored following treatment of exacerbations. We know that there's a poor clinical response sometimes in relation to the antimicrobial resistance patterns that we see in vitro. We know that exacerbations can occur sometimes even when we don't culture CF pathogens on standard culture. And also sometimes there might be a clinical response to antibiotics, but when we monitor bacterial density in our sputum cultures, we don't see any changes there in relation or during exacerbations. So what about the early lung microbiome? The figure on the left is a Kaplan-Meier survival curve, and it shows the proportion who develop bronchiectasis during the first few years of life, cystic fibrosis. And you can see here at around 1.5 years of age, around 50% of infants with CF have some evidence of lung scarring on CT scans. Traditionally, we've been focused on infections with Pseudomonas aeruginosa in CF, and there's no doubt that infection with Pseudomonas is certainly often quite severe and can lead to worse outcomes. But early on in life, other organisms such as Staph aureus and Haemophilus are more common, and so there has been some attention drawn on those. But also, we need to consider the role of viruses, and then, of course, the community of organisms that we don't see on culture, the microbiome. Another recent piece of data that also increases our inquiry about the microbiome are these data that divide the presence of bronchiectasis that occurs early in this Kaplan-Meier curve according to the left lung and the right lung. So what you can see here is that early CF bronchiectasis is much more likely to occur in the right lung compared with the left. However, when we do bronchoalveolar lavage studies, we're much more likely to find infection in the left side of the lung. This is when we consider infection according to standard microbial cultures. So this again raises the possibility that our narrow focus on cultivated organisms may not give us the full answer as to what's going on in early CF. So I'm going to talk about some CF studies from the first few years of life from a number of different cohorts. This is the first study from our group. And this was a study in a cohort of children that were actually recruited in the 1990s. So these subjects are now 25 years of age. They were recruited during an era of newborn screening, but these children did not receive antimicrobial prophylaxis during the first few years of life. So they only received antibiotics if they had acute exacerbations. And the long-term aim of this study is to see if these early microbiome patterns actually have some impact throughout the life course. But in this slide here, what we're looking at individual microbiome diversity in children with cystic fibrosis who are well, children with cystic fibrosis who are unwell, and also healthy controls. And these are infants under 6 months of age. And you can see already that children with CF have a very different microbial pattern within their lung microbiome within the first 6 months of life. And this was significantly different in those who were having exacerbations compared to those who had bronchoalveolar lavage when they were well. So this particular study identified these different patterns early on within the first few months of life. These appear to be mainly driven by staphylococci, and this was before the anti-staphylococcal prophylaxis era, as I've mentioned. There did appear to be a period of very low abundance where cultures were often negative. There was low level of inflammation, and this seemed to suggest that there might be a window within which we could operate to try and impact on this early lung microbiome. When these children got slightly older, we compared their microbiome with the culture results that we got from the standard microbiological laboratory. So in the microbiome, when this was dominated by a certain genus, and by dominance we said that when there was more than 70% dominated by a particular genus, such as Staphylococcus, we found that we also cultured Staphylococci in the Petri dish. So the microbiome and the standard culture in this situation was associated with neutrophilic inflammation in the lower respiratory tract, here demonstrated by the presence of free neutrophil elastase. So what this slide is showing is that when we see some dominance in CF in early life by these specific pathogen-associated genera, we are seeing inflammation, suggesting that there is an association between these microbiome patterns and the presence of early disease. This is from the same study, and these show individual subjects and their data from longitudinal BAL sampling. I'll just take you through a few of these. They're quite interesting. Here's a child who at a year of age has a relatively diverse lung microbiome. This remains diverse at 20 months of age, and then at 47 months of age, this child's lung microbiome is dominated by the presence of Pseudomonas aeruginosa. And similarly, here's a child who has a relatively varied microbiome early on in life at 3 months, and then there's this dominance by Staphylococci. Here's an example which is quite interesting of a child who has a Staphylococci that dominates the lung microbiome at 3 months of age, but subsequently there's a more normal pattern in terms of diversity of the lung microbiome that's restored. And this gives us some optimism again that there may be some opportunities to impact early in CF. So this a couple of next slides show some data from a more modern cohort where antimicrobial prophylaxis was used, and compares the data with cohorts of young children with CF who do not receive antimicrobial prophylaxis. So the top slide are data obtained from oropharyngeal swabs, so predominantly the upper airway, and the bottom slide is from the lung microbiome obtained from bronchoalveolar lavage. And this shows the diversity in children who've not had antibiotics, those who've just had episodic antibiotics with treatment, those who've had antibiotic prophylaxis, and those who've had antibiotic prophylaxis plus occasional treatment for exacerbations. So what you can see is that both in the upper airway microbiome and also in the lower respiratory tract, the antimicrobial prophylaxis is significantly associated with a much lower diversity. You can see that the diversity patterns in the BAL and the oropharynx are actually quite related, suggesting that you can use the oropharynx when you're assessing diversity as a surrogate However, when you actually want to look at the community of species or genera that we're seeing in the lower respiratory tract, they're not directly correlated with the oropharynx. So we also looked at the presence of lower respiratory inflammation for these samples. And this is done by looking at the neutrophil count within the airway and also interleukin-8. And what you can see is that children who had antimicrobial prophylaxis appear to have lower inflammation, which at first inspection would suggest that's very positive. That's an outcome that we would want to achieve in the first few years of life. However, there does appear to be this cost of significantly lower diversity. So what we don't know is that there might be some early gains from early antimicrobial prophylaxis, but there may be a very significant long-term negative impact from diminishing this diversity so early on in life. So the final study from our group in CF was a predominantly cross-sectional study that was done from sampling taken over the first 4 or 5 years of life, looking at the community of organisms in the lower respiratory tract obtained in BAL and using principal component analyses, which identified these 3 clusters, C1, C2, and C3. So C1 was a cluster where there weren't particularly a big biomass of organisms. C2 was a cluster that appeared slightly later in life and was related to organisms that were often found in the oropharynx. And C3 was a cluster that seemed to be more dominated by what we would consider to be pathogenic genera in CF. And that's shown again in some of these slides here. You can see this is cluster 3 in red, which seem to occur in slightly older children. You can see here, this shows the relative abundance of the genera that we're seeing in the microbiome. So the cluster 1, which occurred early, there weren't many copies that were identified, whereas the 2 older clusters, there seem to be a much greater abundance of organisms at this point. And these 2 clusters, C2 and C3, were associated with an increased cell count, neutrophils, and also IL-8, suggesting they're associated with increased inflammation. And they're also associated with some structural changes that are identified on chest CTs. So this C3 cluster seems to be a sort of pathogenic cluster that occurs slightly later and is associated with the early disease outcomes that seem to confer a worse prognosis in CF. So I'm going to go back to the gut microbiome in CF. This was a study that was just done on 7 infants with CF, where I think they used oropharyngeal samples and also sampled the fecal microbiome, obtaining samples in the first month of life and then 3 monthly for about 20 months of life. They showed that there was a core microbiome within the gut, but there were several genera within the gut and the oropharynx that seemed to be shared, and that when there were changes in the gut microbiome, and those changes occurred with some of those things that we talked about earlier in relation to feeding practices such as breastfeeding or the introduction of solid foods during weaning, the changes in the gut microbiome were then also reflected subsequently by changes in the oropharyngeal microbiome, as you can see here for some of these genera here. So, for example, Roseburia, you can see that this increases in the gut microbiome. Initially, there's no change in the oropharyngeal microbiome, but subsequently it mirrors that change. And you can see that's also replicated by these other genera, which has alerted us again to the possibility that we might be able to impact on the lung microbiome in CF by manipulating the gut microbiome. So a final study in CF, of a very recent study published from Seattle by Luke Hoffman's group there, they looked in 200 children with CF and about 35 controls, again at the fecal microbiome, with repeated sampling during the first few years of life. And they showed that children with CF who had low length had a very significantly different microbiome. The first thing, there was a delayed maturation of that microbiome compared to controls, but then there were also some compositional changes. And those compositional changes were manifest with a decrease in bacteroidetes and an increase in the proteobacteria, as represented here in these figures, which is a collation at the different time frames for each of those individual subjects. And that's interesting because when we consider the functional change that this is associated with, this certainly, this sort of pattern of change in the microbiome would predict changes in nutrient harvest and also impacts metabolism of insulin-like growth factor 1. And that also highlights the importance of relating dysbiosis to functional aspects of the microbiome. So finally, I wanted to mention that we are conducting an intervention trial in Melbourne using a probiotic to see if that confers any protective change on the gut microbiome in young children with CF when they're treated with antibiotic prophylaxis and treatment antibiotics during exacerbations. So just to summarize, there are differences very early in the CF lung microbiota, which appear eventually to be characterized by dominance by pathogenic bacteria. These taxa, when identified, are associated with low respiratory inflammation. These taxa are associated with what we culture in the microbiological laboratory. There seem to be very profound effects associated with antimicrobial prophylaxis. There does appear to be a small window in which we might be able to impact the microbiome, and also that there seems to be a rationale for a closer inspection of the gut-lung microbiota axis. So what about atopic diseases? I'm going to present a couple of studies from the COPSAC study now, a fantastic study in Copenhagen, where it's a birth cohort study where children have been followed up till age 7, and all those 700 children that were studied were monitored within clinics associated with that research groups. They had fantastic follow-up right to age 7, where they identified children who had asthma or not using lung function, ISAC questionnaires, etc. In this study, Stockholm measured the fecal microbiome by obtaining samples initially weekly and then 3-monthly during the first year of life. They analyzed samples at, I think they did this initially at 1 week, 1 month, and then at a year of life. They found that the early gut microbiome could be described by 2 components or 2 sort of patterns. But when you looked at children who subsequently had asthma or not, there didn't seem to be any difference in relation to the pattern of dysbiosis that appeared in their gut microbiome early on in life. However, when they focused just on mothers who had asthma, these 2 components seem to differentiate subsequently children who had asthma or not. So it seems that having a mother with asthma conferred high risk, and then these different patterns within the fecal microbiome interacted and subsequently were associated with the asthma phenotype. A more recent study samples from the hypopharynx by using soft nasal catheters to suck out some secretions from the hypopharynx, which they termed as airway samples. And they performed a similar study and identified that at 1 month of age, the microbiome in these samples seemed to again confer an association with subsequent asthma at 7 years of age. And the main compositional differences they identified were in association with Veillonella and Prevotella, but there was also a functional association with the development of an inflammatory phenotype, which is also associated with development of asthma. We've just published, this was published last Wednesday, a study of a birth cohort. And this was predicated on the knowledge that westernized populations have high rates of allergic disease and have been shown to have low carriage of specific taxa within the human gut microbiome, particularly of the genus Prevotella. And Prevotella is one of these organisms that metabolizes short-chain fatty acids and produces propionate. And we think that that has some impact on immune tolerance within the fetus. It certainly has been shown to have that effect in mice models. So we hypothesized that maternal carriage of Prevotella during pregnancy would be associated with the reduction of allergic disease in the offspring. So we obtained fecal samples from women in this birth cohort of 1,000 women at about 28 weeks gestation. We used the 16S RNA gene sequencing, as we described before, in a nested case cohort study. We then determined food allergy status in infants at 1 year of age following skin prick testing. So by food allergy, I mean they actually had food challenges, so we could determine a specific and definitive food allergy in that way. We also reported atopic wheeze. These were children who were sensitized to aeroallergens and whose parents reported that they had wheezing, and also eczema at a similar age. This is a volcano plot that looks at the operational taxonomic units that were identified from the 16S analysis. And it showed that there were 2 OTUs which turned out on PCR to be very, very similar and were speciated as an organism called Prevotella copri, which is one of the Prevotella genus and is an anaerobic organism. And this was the main difference between cases and non-cases. So children who developed food allergy, in their mother's stool at 28 weeks gestation, there it was identified to be this very significantly expressed difference. This is shown again here as the fraction of mothers who carry Prevotella copri, and also the relative abundance of Prevotella copri when it's identified. And here are the children who had no evidence of food allergy at 1 year of age. And you can see that those who had food allergy, the mothers either didn't carry Prevotella copri, or this was identified with much less abundance. And similar outcomes were found when we looked at children who developed atopic wheezing, and also those who developed eczema. And interestingly, similar findings have also been shown in relation to Prevotella, but also this other genus, Lachnospiraceae, in terms of development of infant behavior at 2 years of age, which is really fascinating and highlights again that potential impact on the gut enteric axis in early life. Interestingly, we find that children who have older siblings, children who live on farms, and children whose mother have diets that encourage the growth of Prevotella within their guts had more Prevotella in the maternal stool. And it was the maternal stool that was important because although this was related to the infant and fecal microbiota, it was the maternal stool that predicted these outcomes most strongly. So I've used the term prediction or association because none of the studies I've described can actually be determined to show causality. And I alerted many of you to this fantastic paper in Cell recently that identifies this problem in all of the microbiome research, all the 21,000 studies that have come so far, that there's very few that actually definitively establish causality. Many of you be aware that some of the studies have inoculated mice, experimental mice, human microbiome mice, and look to see whether or not they've developed a sort of human-related phenotype. The vast majority of such studies have had a positive result. And so this paper now analyzes the methodological flaws in such studies and describes a much better way in terms of the experimental detail and also the analytical approaches that we need to do in order to, in the next few years, to establish causality in relation to early microbiome studies. And some of these include different mice models that have a better correlation with the human immune system, using primate models, using new statistical approaches, and also using an interventional framework, using interventions to actually see if we can manipulate the microbiome and change some of the disease outcomes. So this is a complex area. The gut microbiome certainly matters. I've shown a lot of data in relation to the lung microbiome. I think the answer there is that the early lung microbiome probably matters. The lung microbiome is obviously very difficult to study, and we don't have a great surrogate for this in the upper airway samples. But the evolving data do suggest there are patterns of dysbiosis, and this might be similar to those proposed for gut diseases. For example, there are conditions where there is increased abundance of the lung microbiota, and there are dominance by pathogenic bacteria, and cystic fibrosis might be an example of this. There do appear to be microbiome patterns that associated with decreased abundance of health-associated bacteria, such as Prebiotics copri. There are conditions that are associated with delayed maturation, and this was shown in the study by Stockholm et al. And also some aspects of the CF gut studies that I showed earlier too. However, these sorts of patterns of dysbiosis are somewhat simplistic, because there may also be some evidence of increased abundance of pathogenic organisms within the asthma lung. So I would like to thank you for listening to this very complex area. I'm not sure I've done great justice to this particular topic. I hope you can forgive me for that, because the enormous number of papers that have contributed to the literature. But I hope you can watch this space, and Atul, if I've done a reasonable job, please ask me to visit again next year, and I hope we can present some more papers to you all then. Thanks again for listening.
Thank you very much, Sarath. It was an excellent overview, and then you explained in a very simple way so people like me were able to understand it much more clearly. So yes, it will be quite nice for you to come back next year to give us more overview on this changing subject. So the few interesting questions. Could probiotics or prebiotics help in improving microbial diversity in CF or even in asthma?
Yeah, so let's talk about CF first of all. I think the paper by Madan is very interesting. It shows that the changes within the gut microbiome can impact on the oral microbiome. And we do think that the earlier seeding of the lung microbiome comes from the upper airway. So there is a route by which we potentially could manipulate the gut microbiome using probiotics and impact on the lung microbiome. I think apart from studying interventions to look at the causality, I think it's a bit early to be using these interventions, because the studies so far don't actually give us that causality. They show us that some changes in the microbiome, for example, in early asthma, some changes in the infant microbiome are associated with the development of asthma at 7 years of age, but that could be an epiphenomenon. It could be other behavioral changes. It could be related to treatments early on. It could be an impact of viral infections. So we do need to understand a lot more. We need to understand about the function of the microbiome when we're studying it, and we need to understand a bit more about the causality, how they actually cause those phenotypic changes later on in life.
Thank you, Sarath. Tim Newson, what is the impact of CF modulator therapy on gut or respiratory microbiome?
I think we're actually doing some studies to look at that, and I know some people have done some studies using culture as well, and have shown some changes in the organisms that are prevalent in CF. I haven't actually looked at that literature very closely. What we do know is that we haven't, with CFTR modulators, been able to completely reverse chronic infection within the lower respiratory tract. So there is some impact on organisms, but for example, I don't think we've been able to reverse successfully and universally cultures of Pseudomonas in children who are chronically infected. But it's an interesting thing, Tim. I think there'll be a lot of people looking in that area.
Thank you. And we probably, currently we use prophylactic antibiotics. Should we be using probiotic with the patients who are on long-term prophylactic antibiotics with them?
Well, I think probably the first question is, should we be using prophylactic antibiotics at all? We in Australia tend to use them. I know in Europe, in the UK, the majority use them. Our controls for that study came from the US, where very few people would use antibiotic prophylaxis. So I think there's probably equipoise for such a study. I know there are studies like that ongoing. I think that using antibiotics in that way is a very 1980s thing to do. I think we probably need to be a bit clever about how we do them. I think it's fantastic that we've impacted on lower respiratory inflammation by using antibiotic prophylaxis, but it does concern me what we're showing to be the impact in terms of the diversity of the early lung microbiome.
Thank you, Sarath. There's still quite a few questions. I think we ask people to come back next year when they can ask face-to-face questions to you. And hopefully you will be able to join us in London next April